morning, everybody. I am going on to this website that your textbook supports, and I've been using the second edition website, and I opened it up yesterday. I wasn't able to open it up on Wednesday. But for you to navigate, this is where these movies are, and here we are in chapter 11, and that opens up different support figures and topics for that chapter, and process animations. This is what happened last time on Wednesday, but yesterday I had no trouble. So what's with that? I don't think the server's going to help because I didn't go and explore on Wednesday. How about your regular process animations? Okay, so I got through that route. I didn't have any trouble on my computer, so you shouldn't have trouble. Anybody been accessing either the second edition or the third edition of these support material? Hardly anybody. Okay, well, use them. The ones that I cover in class are a little bit redundant from what I cover in class, but a lot of times they are clarifying and good. So it's for your benefit, but there's not too much extra stuff in there. Anyway, that's all up to you. You have these links, they more or less work. And last time we played this, we didn't have the guy. So if he's not there, I will read it to you, because that's all he does, man, is just read that information. Okay, so let's play this.
Influenza A is one of the most common life-threatening viruses in the United States.
Okay.
Each year infecting approximately 10% of the U.S. population and causing about 20,000 deaths. Influenza is normally transmitted by droplets or aerosols from the sneeze or cough of a nearby infected person. When another individual inhales the droplets, the virus has a chance to invade the cells of a new host. Influenza A contains a genome of single-stranded RNA segments, enzymes required for viral replication inside a host cell, and a shell of matrix proteins. The virus also has a phospholipid envelope with several embedded proteins, including a molecule called hemagglutinin, abbreviated HA or H, which plays an essential role in viral cell entry. An influenza virus binds to host cell receptor proteins that contain polysaccharides terminating with sialic acid. A sialic acid that is attached to galactose provides a recognition site for the virus's hemagglutinin protein. The sialic acid can be connected at different positions on galactose. This example shows an alpha-2-3 linkage between the number 2 carbon of sialic acid and the number 3 carbon of galactose. The avian influenza strain H5N1 recognizes mainly this alpha-2-3 linked version of the carbohydrate, which is found in birds. In humans, such carbohydrates are found only deep within the lungs, which may explain why the H5N1 strain, though deadly, is rarely transmitted between humans. In contrast, the upper respiratory tract of humans contains mainly alpha-2-6 linked receptors, and it is to these receptors that human influenza A viruses bind. The hemagglutinin complex consists of a trimer of subunits. Each subunit includes a domain that passes through the viral envelope and a domain that binds to the sialic acid receptors on the host cell. For clarity, let's look at just one of these three subunits. For an influenza virus to become infective, hemagglutinin must be cleaved. An enzyme released from the epithelial lining of the human respiratory tract performs this function. The cleavage frees one end of a segment called a fusion peptide. The fusion peptide is hydrophobic and buried within the core of the hemagglutinin trimeric complex. The host cell takes up the virus by endocytosis. The endocytic vesicle then fuses with a lysosome, and its interior acidifies. The lowered pH induces a conformational change, shifting the receptor binding region back and triggering the fusion peptides forward to penetrate the vesicle membrane. A number of trimeric hemagglutinin molecules in the same region of the membrane mediate fusion between the viral and host membranes. The fusion process expels the contents of the virus into the host cytoplasm. The virus is ready to begin its replication cycle.
Okay, so that is all the material, or that I covered all of that on Wednesday. But I think that movie is, a little, good. Probably better than how I covered it. So there is just kind of supplemental information to clarify. If you have any questions, just go over that, and I think it complements what we did on Wednesday. So that is, the beginning of the coverage of the influenza virus, and I spent a lot of time talking about that initial attachment step because it's so critical in any viral infection. But then we talked about the growth cycle inside the cytoplasm, and tracking a virus needs to replicate its own genome, and it needs to make viral proteins. So track how those two important activities are done in these viral replication cycles. And one way to kind of organize that is that protein replication is done out in the cytoplasm. And typically the viral copying of its genome is done in the nucleus. And so that'll kind of help you organize those growth cycles in those diagrams. So any questions on this attachment and the 2-3? Yes.
Can you just briefly go over the trimer concept?
Yeah. And that's why I thought that the movie did a nice job, because it moved them out of the way. But the two important parts of that trimer in this figure is the carboxyl end, which is the attachment component of the hemagglutinin, and that attaches to the receptor. So that's what is kind of shown in that circular. And then there's this cleavage event that then allows this hydrophobic region, this fusion component of the hemagglutinin, to move forward and then fuse, just like the word says, fuse that that virus to the outer surface and then release it. Yeah, so that's two different events. So the attachment is to the outside of the cell, where the carboxyl terminus is involved in that attachment. Once it's inside the cell, now you have this fusion event, fusing the virus with that vesicle and then releasing it. So it's a good question, because it's a two-step process, because there's two domains that are involved in this viral invasion. Other questions about influenza? Okay, so we're going to do one more virus, and that is the human immunodeficiency. And an example of a retrovirus, because of this reverse transcriptase enzyme that has to go in reverse before it can ever go forward. And so, they're at the top. talking about it has an RNA genome, and the first thing it does is make double-stranded DNA. And why? Because it gets integrated into the host chromosome. And there the virus can remain latent, hidden, without any symptoms, just that viral genome is copied. Anytime our host, anytime we copy our DNA, we're copying the viral DNA, kind of unbeknownst to us. And it has this term, this whole group of retroviruses are called lentiviruses, from the Latin, or at least the Spanish, lentos, meaning slow. So they slowly develop over years their symptoms. So simple retroviruses are causing tumors, causing leukemia. The lentiviruses, which is the HIV, is more complex, and this is the slowly exerting its symptoms over years. Okay, and the idea is that this was originally an SIV, simian influenza virus, in Africa in the early 1900s. There's been some interesting autopsies of monkeys from museums, monkey specimens that have recovered HIV-like viruses, and so kind of patching together kind of the recent history, is that where HIV developed from? Well, there's evidence to support that. Okay, so the disease that it causes in people is acquired immunodeficiency syndrome, transmitted through blood, genital, oral genital contact. So it's a sexually transmitted disease. No vaccine or cure at this point. There certainly are drugs that buy patients, in some cases, a lot of time. So there's some intervention, but it's extending life expectancy. Okay, so let's take a look at the structure of the HIV is similar to the structure of the influenza virus. So kind of starting with the innermost, here is its single-stranded RNA surrounded by, again, the first layer of protein, from the inside, surrounded by a little protein coat called a nucleocapsid. And then kind of working your way out from that, there's another protein coat. This is kind of an unusual. This kind of yellowish structure is the capsid. And so that's the second layer of protein. And then moving outside from that is the third layer of protein. This thing is really protected by 3 layers of protein, is the matrix protein. And then outside of that is the lipid bilayer. Okay? So, track those 3 layers of protein in HIV, which I think that's one more than the influenza A virus. I don't think the influenza A virus has that inner matrix protein. Yeah, so you see it just has a nucleocapsid and an outer matrix, and HIV has a third layer of protein. This yellowish, this capsid. Okay, and similarly to the influenza virus are these glycoproteins on the outside, and we'll talk about them. Now, also similar to the influenza A virus, it carries its own proteins, and so the protein that HIV carries is this reverse transcriptase, or at least that's the one that I want to focus on, and that's shown kind of in this reddish-orange, because this is the special polymerase that takes RNA and makes double-stranded DNA from reverse transcriptase. Very unusual enzyme. So, of course, that's not in the host, and so these viruses have to carry their own special enzymes to get their system started. So that's kind of material. So we're going to be talking about the spike membrane protein that attaches the virus to the host cell. That's always such an important step. Okay, so in the bottom part of this figure is then the genome of the HIV. And it's kind of an interesting figure. It shows you where a number of these proteins come from. So the GAG proteins make the capsid protein coat, as well as the matrix protein. And so that one gene is involved in is the main production of these protein coats that are involved in HIV. And then here's the pol genes, which are going to synthesize polymerase genes. So the reverse transcriptase is right there. So just, and then the glycoproteins, the envelope proteins. But what's interesting, and is a good example, is there's a few things going on. You can see these genes overlap with each other. So the end of the GAG genes extends through the beginning of the polymerase genes. And you can see a number of these genes overlapping. And do I say anything about that? Yeah, viral efficiency. Genes are overlapping, so they're saving space. These are small genomes, but they're getting extra duty out of that same DNA sequence, either by transcribing in the opposite direction for one gene, and then vice versa for the other gene, or simply changing reading frames. If you change reading frames, you're going to make a completely different protein. So this is one of the things viruses are famous for, is making do with very little. Very small viral genomes pack a wallop. Okay, so similar to what we did with the influenza virus, here is attachment according to the HIV scheme. And there's some things a little bit in common. So what's really important is that we didn't stress with the influenza. No, we did. You know, the receptor-ligand interaction is really important. So the receptor, in the case of HIV, is this CD4 protein, and it's typically I'm pretty sure there's a carbohydrate, a glycoprotein, and then the CCR5. Why that is important is because, and I should put this in, these are only present on T cells. This then defines the host specificity. The CD4 and CCR5 only present on the immune cells, T lymphocytes. And so that's why it attacks our immune system. It doesn't, you know, it's not, so not only is it specific for the human, it's specific for only those cells in the body that have those receptors. And it's just a subset of our immune system, and that's the T cells that have these receptors. So that explains why immunodeficiency is because it's attacking an important group of T cells in the human. Okay, so here's the glycoprotein on the HIV virus, and glycoprotein 120, GP120, is the subunit that binds this CCR5 CD4 co-receptor. Okay, it actually needs both receptors to lock in, and that then causes this transformation. And why do I have that kind of overlapping? Sorry. Where am I? This is, I think I moved this. Okay, causes this fusion event between the viral surface and the human cell, and then the HIV core enters the cytoplasm, releases the viral RNA, and reverse transcriptase. Okay, so there is your initial attachment step using that glycoprotein GP120 and then CCR5 CD4. Okay, so what does that reverse transcriptase do? It copies that single-stranded RNA to double-stranded DNA, and it actually has 3 different enzymatic activities. It has this reverse transcriptase, where DNA is synthesized from this RNA template. But it also has an RNase activity, meaning it degrades RNA. And then thirdly, reverse transcriptase. Now, I'm just going to correct this. This is a little redundant. DNA synthesis from the RNA template is synthesis of double-stranded DNA. And so, let's just go with 2 activities. Excuse me. So I'll go look and make sure this is correct, but at this point, I just don't see any different. DNA synthesis from RNA template, that is step 3. So let's just go with 2 different activities. And I'm not even stressing the RNase activity. But for your interest, it has more than one activity. The main one I'm stressing is this reverse transcriptase. Okay. Now, what's interesting about viruses in general, and HIV in particular, is this this high error rate. So the polymerase that does simply copy the RNA strand to make more RNA genome has a high error rate. It does not have proofreading capability like our DNA polymerase III has, and so it makes a lot of mistakes, introduces a lot of mutations, and guess what? Stays ahead of our immune response. So in one instance, in 1 week, we develop an immune response to HIV. The next week, it's made a few more mistakes, and now it's a different virus. These mutations are introduced on a pretty regular basis and introduced rapidly. It has a high mutation rate because of the lack of proofreading. And so it's kind of part of its virulence. So that's generally true of a lot of viruses. Their polymerases introduce mutations, and especially true of HIV. Okay, so like we've said, attachment is the first step, so we've already looked at this step here, looking at the CD4, CCR5 co-receptors, which are only present on T cells. And you now have introduction of the capsid and then release of the RNA reverse transcriptase, and your negative strand of DNA is then synthesized by the reverse transcriptase into double-stranded DNA. And now, just to say what happens to it, it gets incorporated into the host chromosome and sits there and gets copied by the host cell over time. Somebody said, well, asked me, what causes it to come out of the chromosome? It's a good question. Those signals are being identified. I don't know that answer, but there's differences in cytoplasmic chemistry that cause it to come out of the host chromosome and start expressing viral genes. So when it does come out in the cytoplasm, so you can see now host ribosomes, which are shown in red, are translating now viral proteins. And so here are these protein coats, this GAG protein that is shown here, then this GAG pol precursor. So remember, the polymerase and the GAG genes overlap, and so that is synthesized in one big strand. It then is cleaved, which is actually not shown. And then you get aggregation of the proteins out in the cytoplasm, kind of putting a coat back on the viral genome. And then, over here on the ER is where these glycoproteins are synthesized and inserted in membranes. And so the membrane synthesis is going on typically on the Golgi apparatus and the ER. And so here's the synthesis of the membrane coat, which then actually gets incorporated into the membrane of the cell, so that when the viral genome has been encapsulated by the capsid proteins, when it buds out from the cell, it is budding out and bringing with it those glycoproteins, those viral glycoproteins, which allow it to attach to the next set of cells. One of the really interesting things that happens in HIV is, and it's shown here is a virus inside one cell that is fusing with another neighboring mammalian cell. And so there's direct passage of a virus from one cell to another without being released out into the bloodstream, without being released outside the cell. And why is that important? Because there are antibodies outside that cell waiting for it to destroy that HIV antigen. And here's this direct passage from one cytoplasm to another cytoplasm and escaping the immune system. And so right here, this is really an important mechanism by which HIV is transmitted. Yes, some are released into the bloodstream and circulate, but some go directly from one cell to another, which is part of how it escapes immune detection. One other interesting point is this slide, which, there's some reference to this tetherin protein that keeps these glycoproteins and the virus tethered to the cell. And so here's a nice transmission electron micrographic side view of HIV still tethered to the host cell that it has invaded and is being released from. So that's just kind of a blow-up of what we just saw. But here's this alternative route. Again, no release of HIV outside of the cell, and this is pretty new, that these nanotubes, so this is one mammalian cell, and here's another mammalian cell just stained with different dyes, but showing that this cell is extending an HIV-laden nanotube, and this cell is now being infected by this cell through this unusual structure that, again, allows the HIV to avoid detection outside those cells. So it actually has two different mechanisms to evade the immune system. Really, it's a very, very tough virus to deal with. The couple of combinations of the high mutation rate and then this immune evasion. And then the fact that it's attacking the immune cells themselves. So it's killing the immune system that would otherwise kill itself. So it's just an extremely potent, difficult virus to deal with. So that's the coverage of the HIV. Any questions on HIV? Any aspect of that? Yes.
I'm wondering if do all the HIV cells escape the blood, or do some of them actually get caught by the defense mechanism?
Yeah. There is a whole series of battles going on, and HIV gets consumed, HIV gets phagocytized and destroyed, and it's just in the end the HIV slowly overwhelms the immune system. But there are battles won, but the war in the end is HIV's. Other questions? I will clarify that point about the roles at least for the purpose of the class, the main role I'm interested in reverse transcriptase is going from single-stranded RNA to double-stranded DNA. Okay. Then I'm just going to finish up this chapter looking at, these are kind of unusual features of our chromosome that look like remnants of long time ago retroviral infection that we're still carrying with us. And it's incredible, high percentage of our genome are not coding for human proteins. They're not coding for any proteins. They just look like they're remnants of ancient viral infections. So they're called retroelements. And it's a little bit of an interpretation. Are these retroviruses, I should say viruses, that became trapped in our genome? So that's a question. Yeah, make that a question. Are these retroviruses that became trapped in our genome? About 50% of our genes have these traces of retroviruses. And so some of the remnants, you can see these GAG genes, these polymerase genes, these viral envelope genes, and it kind of shows the different kind of forms of these what looks like functionalist genes, because if any genes they're coding for, they look like viral genes. So just an interesting feature of our chromosome are these retroviral segments. Okay, and then the last thing is this really interesting, can we make use of these HIV-like viruses, these lentiviruses, in gene therapy, in cancer therapies? And the answer is yes. So really an interesting application of some cutting-edge biomedical work. Okay, so you need a vector if you're going to do any sort of gene therapy. The context of this is genetic defects potentially can be repaired with new genes. And so how do you bring those genes on site into your liver or into the organ that has this genetic defect? Well, you can modify a virus to go directly to that cell type because their ligand on their surface will only bind to that particular organ. So we've seen a number of examples of that. The flu virus only infecting the lung because that's where the receptor is there. The HIV virus only infecting T cells. So you have the really neat specificity that viruses allow. And then incorporate a few enzymes, and we'll look at exactly how an HIV has been dismantled and then rebuilt into something that can work for us. So originally this work was done with adenoviruses, which were directly double-stranded DNA viruses, because in the end, you need a double-stranded DNA to do any sort of gene therapy. We are double-stranded DNA, and so whatever vector you deliver needs to have double-stranded DNA. So that's why adenoviruses were first used, because they are a double-stranded DNA. But more successful has been this single-stranded RNA using reverse transcriptase and making the double-stranded DNA, and using this whole lentivirus approach that HIV uses. So they're called lentiviral vectors, or even lenta vectors. And so here's a figure that's new in your third edition, not in second edition. And so this shows a genetically modified HIV virus. And one change that's been made, and I mean it's completely dismantled. So it's interesting. I don't know why I'm not covering the steps of that. But there are. It does show in the end of this chapter the steps that are required to remove the virulence genes and replace them. But here's one example. Instead of GP120 on the cell surface of the HIV, you now have a completely different envelope protein that then is tailored to wherever in the body you need to bring this, this new gene in. And in this example, it's much broader specificity, so it can go to a lot of parts of the body because you have engineered a new glycoprotein to attach not to T cells, but to where you need it. So it actually does have this reverse transcriptase. And so, look inside here. It looks like the matrix protein, the capsid protein, and nucleocapsid. 3 layers of protein. It has reverse transcriptase. It has components of the HIV. And so once the RNA hits the cytoplasm of the cell, it is synthesized from single-stranded to double-stranded. Now, what's of course shown inserted in this single-stranded RNA is whatever gene therapy, the healthy gene that it's replacing in the gene in the patient that has this genetic disease. So here is an inserted human gene in what would otherwise be viral sequences, and it goes through the same system where it's synthesized into double-stranded DNA, and then incorporates. It goes into the cytoplasm, and because of the insertion sequence, incorporates right into the human chromosome. So we're capitalizing on HIV's normal progression, but using it in a therapeutic setting. So really neat example of redeeming and capitalizing and using some ingenuity to make what had been an enemy now work for us. What's really important then is how do you get this particular gene, this healthy gene, to express its healthy protein. So that's another layer of the therapy that's a challenge, is to get that in situ expression of that gene and at the right time. So it's complicated, but one cool thing is here's a photograph of a child that has recovered from a genetic defect because of this therapy. I think it's the first patient that has been successful. So, hey, this is not in the textbooks, or it's not in theory, it's being used. Yes?
A question, could they also do that for individuals that have a genetic disorder that, for instance, in their chromosome number 7, they're missing genes, parts of their genes, and would they be able to insert that as well?
I think that, a lot of, I don't know that particular gene, but if there's genes, portions of genes missing, yes, in theory, this therapy can work. There's a lot of complicating things going on, and I just mentioned one of them. How do you get that protein expressed in the target cells? How can you target those cells that need it and those that don't? So it's slowly being developed in terms of an effective therapy. And it's going to be years as it unfolds the technology to match the disease. So I'd say in theory, yes, and it's always complicated. Other questions? Okay, so just a couple of caveats, and in terms of a little bit of the steps that have to be taken to use these viral vectors. Gene therapy is approved only for conditions that are life-threatening. So I'm not sure if what you were asking about was a life-threatening disease, but right now the laws are saying, hey, this is a risky therapy, and so we are just using this in life-threatening conditions. And
For instance, this individual has health issues, heart condition, kidney conditions, medical
Sure, sure.
conditions, and I was curious if this process could help someone.
Yeah, yeah. And I'd say it's being developed towards that broader application. But because it's a risky therapy right now, there is some laws in place that say, some people's the lives have to be threatened for these, and other therapies inadequate. So like I said, they've been engineered to decrease health risks, deleting the virulence genes. Evidently, if you insert next to an oncogene, which is the genes that we harbor, that if they are expressed, can cause cancer. So, cancer is such an interesting biological problem. Viruses can cause cancer, but our own genes can cause cancer. So oncogenes are these genes that are either defective or under some conditions are expressed and cause cancer. And so you don't want to mess with the oncogenes. You don't want to have an insertion near an oncogene that then can activate it. Altering tissue specificity, I've talked about already. And then there are rules in place that say, no, don't alter your sperm or egg cells and then carry this genetic modification on to the next generation. So there is legislation right now that is keeping it at bay because it's still therapeutic. It is, or I should say, it's still experimental and risky. And so that's just some of the caveats that are covered at the end of the chapter about this really neat viral vector gene therapy approach. Okay, it's also used in cancer therapy. You can deliver toxins directly to cancer cells because you can modify the ligand that's on that cell